Bismillahirrahmanirrahim. Assalamu alaikum everyone. My name is Noor Shehbaz. I am from Department of BS Biotechnology and my ID is 009. Today my presentation topic is cotton curl leaf virus. What is cotton curl leaf virus? Cotton leaf curl viruses are a number of plant pathogenic virus species of the family Geminiviridae. In Asia and Africa, the major disease of cotton is caused by the cotton leaf curl Gemini virus. Leaves of infected cotton curl upward and bear leaf-like initiations on the underside along with vein thickening. Plants infected early in the season are stunted and yield is reduced drastically. This virus devastated the Pakistan cotton industry in early 1990s, where it caused an estimated yield reduction of 30 to 35%. Scientific classification of cotton leaf curl viruses. Virus rearm Monodnaviria, Kingdom Shotokiovira, Phylum Cresnaviricota, Class Ripansiviridae, Order Japalafuvirus, Family Geminiviridae, and genus is Bigomovirus. Description of cotton leaf curl disease. All bigomovirus species causing cotton leaf curl disease have geminate particles approximately 18 to 20 nanometer in diameter and 30 nanometer long and a circular, single-stranded DNA genome. All except cotton leaf curl crumple virus have a monopartite genome, with all particle products required for replication, systematic movement, and whitefly transmission encoded on a single DNA component of 2.75 kb. The genome of cotton leaf curl virus is bipartite. Two smaller circular single-stranded DNA molecules named DNA1 and DNA beta are associated with the range of monopartite bigomoviruses from the Old World, including the cotton leaf curl viruses. These molecules are recorded as satellite molecules as they depend on the helper bigomovirus to support one or more stages of their infection cycle, movement and insect transmission for both molecules, and additionally replication in the case of DNA beta. DNA beta is symptom modulating and typical cotton leaf curl disease symptoms only develop when this molecule is present. In the absence of DNA beta, the concentration of viral DNA is low and the symptoms of infection very mild. DNA beta has a single open reading frame which encodes a suppressor of RNA silencing. DNA1 is homologous to the DNA-R component of the nanoviruses and encodes a master replication initiator protein, but its presence does not alter symptom expression. The plant resistance to against cotton leaf curl virus was described by Ali 1997 to 1999 that the cotton curl virus resistance was controlled by single dominant gene and could be transferred to any cultivar by using backcross technique. How does it spread? Cotton leaf curl disease is spread between plants and Bemisia tabaki whitefly, and movement of the insect can rapidly spread the disease through a crop and over large distances. Alternative hosts such as okra, cowpea, radish, tobacco, tomato, French bean, chili, hibiscus species and papaya may act as reservoirs for the pathogens during and between cotton crops. These plant species are cotton leaf curl disease, can be graft transmitted, but is not mechanically or seed transmitted. Where is it now? Cotton leaf curl disease has been reported in Pakistan, India, Egypt, Nigeria, Tanzania, and Sudan, but the viruses and DNA satellite vary by region. Neither the viruses nor the DNA beta satellites are known to occur in Australia. Diagnosis, monitoring, and transmission characteristics of cotton leaf curl virus. Cotton leaf curl disease, a major threat to the cotton industry, has a complex etiology. Despite the identification and involvement of cotton leaf curl virus and additional different satellite DNA molecules termed as DNA beta and DNA1 with cotton leaf curl disease, the precise causative agents appear to be nuclear. A cotton plant exhibiting symptoms of cotton leaf curl disease was collected from Punjab and investigated for the presence of cotton leaf curl virus in its host and vector. It was transmissible through whitefly, causing cotton leaf curl disease. Symptoms in cotton implying virus-specific primers located in the conserved regions of the coat protein gene. The total isolated from infected tissues of cotton leaf was subjected to PCR. It yielded a DNA fragment of above 550 base pairs, which was cloned and sequenced in multiple sequence alignment with other cotton leaf curl virus isolates. It revealed up to 99% nucleotide sequence identity in the corresponding regions. PCR and southern hybridization assays were applied to monitor cotton leaf curl virus in its host and to study transmission characteristics. A time course experiment demonstrated the presence of cotton leaf curl virus in its alternate host 12 days before the appearance of symptoms. Further, it could be detected in a single whitefly vector. Transmission studies showed the cotton leaf curl virus should be acquired by whitefly within age and was transmitted to test plant within 1 hour of feeding. 2 whitefly were sufficient to induce symptoms. This study will help in understanding the epidemiology of cotton leaf curl virus and designing effective management strategies to control this devastating disease of national importance. Now, what caused it? Symptoms are caused by the cotton leaf curl virus, which is primarily transmitted through white flies. The spread of the disease is in part determined by prevailing winds, which will indicate how far the white flies can travel. White flies are most problematic in the mid to late season. The disease is also associated with shelters such as trees. Since the disease is not seed-borne, the virus persists in the landscape via alternative hosts such as tobacco and tomato and weeds. Some additional factors that can favor development of the disease are recent rainfall, infected transplants, and the presence of weeds. The virus is most likely to spread under a temperature range of 25 to 30 degrees Celsius. In nurseries, cotton plants are most prone to infection during the seedling and vegetative stages. Symptoms of cotton leaf curl virus. The primary symptoms of cotton leaf curl virus is the upward curling of leaves. Additionally, leaf veins can thicken and darken, and outgrowths, inactions, may form on the undersides of leaves, typically in the shape of leaves. Flowers may stay closed and then drop along with the bolls. If plants are infected early in the season, their growth will be stunted and yield will be reduced significantly. Now, the last topic of my presentation is prevention and control of cotton leaf curl virus. Number 1. Use resistant or tolerant cultivars. Number 2. Protect seedlings from whiteflies. Number 3. Use only good seeds and healthy transplants. Number 4. Control whiteflies. Number 5. Immediately remove infected looking plants and bury them. Number 6. Control weeds. Number 7. Do not plant cotton near tomato and or other crops susceptible to whiteflies or lice. Number 8. Use acephate imidacloprid at 50% to 1.8% respectively at every 7 days. Plow under all plants debris after harvest or burn them when possible. And number 10 is practice crop rotation by planting crops that are not susceptible to whitefly. Thank you.